destroys the myelin. Okay, question 3. Multiple sclerosis is a neurological condition in which the body's immune system destroys the myelin sheath that surrounds and insulates nerve axons. A clinical study was carried out into the effects of a new drug, interferon beta-1b, for this condition. A randomised trial with a negative control group, placebo, was carried out across 4 different health centres. During the study, patients were given one of 3 treatments, either 0.00 mg, which is the placebo, 0.05 mg, or 0.25. The patients administered the drug themselves at home. The study measured how effective the drug was by asking patients to record any worsening of symptoms after 2 years of treatment. The study involved 372 patients aged 18 to 50 years. 14 patients dropped out before completing the trial. Patients' results are shown in Table 1. Okay, so in Table 1 we have the level of the interferon beta-1b, obviously your 0.05 and your 0.25, and the proportion of patients reporting no worsening of symptoms. We have 16 at the placebo, and then you've got 18 and 25 at the next levels. At one of the health centres, 52 patients were MRI scanned every 6 weeks to monitor any new damage to nerve tissue, results shown in Table 2. So you've got the level of the drug again, and the proportion of patients showing new nerve damage, 29 in the placebo, nothing recorded for 0.05, and 6 at 0.25. Okay. Identify the independent variable in this trial, so the thing that was changed. It is the level of interferon beta-1b in treatment. I personally would just lift the entire thing out of the heading. It's a nice easy way to be sure you've got everything. Okay, trial was carried out in vivo. State one advantage of this type of trial. So basically we are doing this in living tissue, so in a whole human, rather than trying to do it in vitro, in culture. So, well, for a start, culture is difficult for all sorts of tissue, definitely for nerve tissue, so you could go down that route. But what really you're more likely to do, anything that's about in vivo, what you're saying there is that you can see the effect on the whole organism. So that means that you're looking at not just at the specific cells, but every single potential side effect. And if you see that, then you've covered all of that thing. Okay, I put this up the way to try and get everything side by side. For C, explain why a placebo group was included in this trial. So this is basically saying, can you explain exactly what a control is for? So in this case, what we're looking at is to allow comparison between no drug and getting drug. So what you're trying to do is just remove that effect to check that it is the drug that is giving the impact that you're seeing. Okay, and you can see that lots of different ways. Okay, suggest one way in which the results of the trial may not be reliable. Okay, there are several things in here that you could pick out, but let's just start at the top of the statement. Okay, clinical trial was carried out into the effects of a new drug. That's fine. Randomized trial. That's good. Negative control group. Also good. 4 different health centres, that's good, okay. 1 of 3 treatments, this is all fine. And then we hit the patients administer the drug themselves at home. Now that could be an issue, because that is requiring this exactly the same thing to be happening with lots of people who are not under supervision. Okay, that you could put that one in. The study measured how effective the drug was by asking patients to record any worsening of symptoms. Well, this is also an issue. Because if you're asking people to record how they feel, you've got a real difficulty of perception kicking in there, and that might not be reliable. Study involved 372 patients. This is not a massive number. Okay, so again, sample size you could say for that one. And we've got 14 patients dropped out. So you don't know why they dropped out. You don't know if it's because they had a huge worsening of their symptoms, in which case that would have skewed the results. So even just up there, you've got issues with that. The table 1, I don't think gives you any problems, but table 2, this no data recorded here, means that it's very difficult to say exactly what was going on between the 0 and the 0.25. And again, this is a small sample. So any one of these things picked out would be perfectly reasonable to put in. Okay. Describe an ethical issue that the researchers would need to consider before this trial. The main one that you'll see, and I think basically if you bring this one in each time you're okay, informed consent. Right, that is difficult to truly make sure that you have given consent because you properly understood everything that was going on. Okay, so informed consent is a huge one and absolutely one of the easiest ones to just give when you're looking for an ethical issue. And then you've also got the right to withdraw is essential, and a huge one would be confidentiality of data. So everything about you, about what you've done in the study, and also your data itself, how that is used, is it properly anonymised before it goes to anyone else? Big things about that. Suggest 2 conclusions that can be drawn from the results of this trial. Okay, well, I think the best one to look for, or one of the, is definitely in the table. So I would look at 2 different ones. Okay, this one here tells us that 16% on the placebo said there was a worsening of symptoms. But then as you go here, we see that with the drug, there is more of them who are saying they're not, sorry, did I say no reporting of symptoms? 16% say no worsening. 18 to 25% are saying no worsening with the treatment. So you could just go with a straight, having the drug, you prevents worsening of symptoms, and that would get you that 1 mark, but it's worth 2. So what you can then say is that increasing the level of the drug, because there's 0.05 to 0.25, and that's increasing it even more. So not only is it just for conclusion 1, you can just say that you've got the fact that it is preventing worsening of symptoms, and then for conclusion 2, that increasing the amount of the drug actually increases that even more. Okay, so you've got more of an impact with increasing concentration. And that would give you 2. And then the other one is to go to the second table with this one, proportion of patients showing new nerve damage. And since we've got 29 here and 6 here, you can see that the drug prevents new nerve damage from the MRIs. Okay, so that's pretty much the only conclusions that you can get from the data that's there. Any 2 of them. And that's the question.